Hi, everybody. I'm Tal Franji from Immunai, where I work at the software systems team. Immunai's goal is to reprogram the immune system. We detect more than 50 types of immune cells, and we study how they function. We use these insights to help create new treatments or drugs. Our research pipeline starts at the lab, where preparing blood tissues or blood or some tissue samples for sequencing. We use a process called single-cell RNA sequencing, which means we get a stack dump of all the active genes for each one of the immune cells in the sample. We use the data from sequencing the cells for each patient at each time point, combined with data about their condition and treatments to reach scientific discoveries. What I want to present today is how we manage thousands of large files with very complex pipeline in the cloud storage. I'll start by describing the cloud storage. Most companies have moved to use cloud-based storage and have forfeit any use of a physical disk or physical machines. Cloud storage with similar feature is offered by all major cloud providers. However, there are some unique features of cloud storage which makes them different from disk files or network file systems like NetApps. These cloud storage are large key value store. Each value can be up to a few terabytes. They have good reliability and a backup. They can scale and allow access from global locations with low delay. They can be used as content distribution network, for example, for static web images. And they have price tiers. They allow you to pay less for data you access less. One thing to note is that there are no directories. You cannot do operations on groups of values like you do in file system. Our genomic pipeline is fairly complex and contains several stages with different data types, which will not present at this moment. Each set of samples from the clinical study can result in running hundreds of Docker containers. The actual logic that runs inside a container is written either in R or in Python or a combination. All the orchestration logic and the API to manage this processing are written as Google Cloud functions, which are similar to Amazon Lambda and are written in Python. Each pipeline step includes several containers running. Each step depends on the output of previous steps. Each container processes a set of files and creates output files, which is natural. We sometimes need to rerun some of the steps because of changes in the lab process or different quality measurements for specific sample. Our abstraction of each container's inputs and outputs is what we call a data set. The actual data of the data set is diverse and depends on the type of calculation made and the tools used. Each process also creates metadata with the data set. Metadata includes attributes about the lab process, about the computational biology attributes such as cell counts and contaminations, and also processing attributes such as the Git branch used for the code that does a calculation. What researchers want to do? Researchers want to be able to find data sets and process them according to different labels. For example, find all data set of type cell count from the projects arm A and arm B, experiment arm A and experiment arm B for a specific pharma company with a specific treatment with the label enriched T cells. So you can ask, why not just use files, right? Every data set can be kept in a directory. So even with normal file system, using the file tree to index and find files is not convenient. When indexing is done on several accesses, you need more than one dimension to find the data you need. Cloud storage are even less flexible than normal file system, as you can not walk the directory tree. So cloud or blob store are not a file system. It is a key value store. Using the directory tree as index is very slow. Some blob stores also have support for metadata per file or object, but changing them or indexing by them is slow. So how did we implement data sets? Data sets we store data sets as records in a Postgres database. Each record holds the file location in the blob store and semi-structured metadata. We use Postgres JSON-B data type, and Postgres is very good at indexing this JSON-B object and allows to query about attributes from that metadata. We've created an API to find data sets according to different labels. The API returns a data frame with a list of relevant data sets, which is an API that is familiar to our data scientists and computational biologists. In this example, I use Python API. We also have a similar R API to get a list of data set according to some metadata parameters, the type of the data set and the batch of sample processed. In this example, single sample analysis, data sets created after our pipeline separates all the mixed samples and all the cells in them into something called gene metrics. The data frame returned includes metadata from the fields, which are included in the JSON-B metadata that was stored with the data set. This is the rest of the example, and the flow the general flow is query the metadata to get the data set you need, take their IDs, and use the API to get the actual data from each of the data sets. Then in this pseudocode example, we do some clustering over the cells, which will help us later annotate or group them according to the cell type. So the first step of the API is describe data sets, which calls Google Cloud function through a REST API, and which returns the metadata for the requested data sets. After we have a list of IDs, we use the API multiple times to get the data for the data set. The API first calls the server to get the file location, and then calls the Google Store API to get the actual data from the blob store, from the cloud store. The reason for not having the server get you the actual data is that we want to use the Google Store API for two reasons. One is that it's better at bringing large data sets or large files, and the other one is we want to use the user credential, the user or the machine that's running this program. We want to use his credentials for accessing the Google Blob Store. After the processing the API, what we do is we file the data. We first upload the result data sets into the cloud store. And then we use our API to file the metadata with the location of the file in our database. So the idea of having a combination of large file system, large system for large files like Google Cloud Store or file system like HDFS is not new. Apache Spark uses a similar combination. It uses an RDBMS for the metadata stored in some kind of database, whether it's Derby or Oracle or other, and the actual data for the tables is stored either in HDFS or S3 or Google Storage. This is very common in big data because we want to access the metadata quickly, while the large files are accessed slowly. So this was our presentation. If you have any questions, I'm here to answer. Thank you very much, and hope you enjoyed.